And welcome to today's webinar on how to measure water activity in 1 minute. Today's presentation will be 20 minutes followed by 10 minutes of Q&A. If you have a question during the webinar, type it into the questions pane. You can submit a question at any time and we'll be keeping track of the questions submitted to answer during the Q&A. To answer our most commonly asked question in advance, yes, we will be sending out links to the on-demand webinar and the slides to everyone who attended today's webinar. Those will be available a day or two following the webinar. Now I'm going to hand the microphone to Mary Galloway and Dr. Zachary Cartwright, both food scientists here at Meter Group, who will be today's presenters.
All right, thank you for that introduction, Brad. Mary and I are really happy to be here today and excited to be talking to you about this new technology that we've been working on at Meter Group. So the first water activity meter was released back in the 60s, and this meter took about 60 minutes to get a single reading. So this meter used a secondary method that changes the conductance of the sensor. And then back in 1987, Meter Group, which was formerly Decagon, came out with a primary method using a dew point sensor to reduce the 60-minute time all the way down to 5 minutes. So that means within the same time period that you could get 12 readings for the same batch using this method. And then today in 2020, here we are with a 1 minute water activity reading, and Mary is going to go into the details on exactly how this is being done. So for the contents today, Mary will be starting us off by talking about the science behind water activity measurements, and then she'll review some of the ways that these measurements can be sped up. Then I will be talking about using speed to reduce the uncertainty in these water activity measurements and talking about some of the business impacts that fast water activity has already been having for some early adopters of this technology. Then we'll finish with some key takeaways for today's presentation and then move into our question and answer section. So I'm going to go ahead and turn this over to Mary so that she can talk about the science behind water activity measurements.
Thanks, Zachary. All right, let's get into it. All right, we're going to look at this from a physics perspective. So water activity is a very complex process, and there are actually several first order processes that are going on, all with a different time constant. So this is just kind of a little graphic here, giving an example of what I'm talking up here about. And they have to do with the sample, the chamber, and the sensor. So there's lots of things going on with the sample. We have the temperature of the sample. We also have how the water vapor is within the sample, how equilibrated that is, how it can release. With the chamber, we have vapor pressure and also a temperature issue, as water activity is a temperature dependent measurement that has to be measured very accurately. And then with the sensor itself, we have the humidity of the sensor, and especially for those secondary methods, the humidity in the chamber has to penetrate into the sensor, and so that it will change that conductive property, and then that is how they measure water activity. So not only are these time constants all different from one another, they are different for each product and can change under different conditions. So I will talk about that a little bit more with some actual examples as we go along here. So what are some keys to speed? How can we speed up? We've got all these processes happening. How can we make it move faster? So first, we're going to take the sample. So as I mentioned before, we have that water vapor within the sample, and it needs to come to equilibration. It needs to be able to be released into the chamber. So we can help speed this up by doing a nice sample prep. So by cutting or crushing, especially with some samples that have like a coating on it, we need to break that up so that we can get that moisture to release. And then also the sample temperature needs to be the same as that testing chamber. So we can use temperature equilibration trays. We can set that on a temperature plate and get the sample to the right temperature before we even take the reading. Next is helping with the chamber. So once that sample releases that moisture, it has to equilibrate into the chamber. So it's like a second stage there. So if we have a fan within the chamber, we can help with that process, help that happen. We can also reduce the volume that that vapor has to inhabit by making the chamber smaller. And then lastly, we can have non-absorptive surfaces. So this can be debris, but it also can be like a coating. We found in some studies that we've done in the lab where a more of a rougher coating has really slowed down our water activity readings because there's more surface area created, and that surface area also has to equilibrate. Everything kind of has to be in equilibrium there. And then lastly, we have the sensor. So we can improve sensor response time by like using firmware and that type of thing to speed up its responses, or we can just source a faster sensor. So what do you do after you've done all of these things and you still want a faster reading? You can use a predictive model. So we're going to talk about that. So first is a first order model here. And it's a well-known engineering strategy. And basically, you get empirical data, you get actual real data, and then you fit a mathematical model to it. And you can speed up this process because you're basically predicting where you think it's going to end. So there are some problems with this type of just first order modeling. For one thing, if you're using the reading time series data, it obscures some of the processes. When we look at this time constant, this rate of change and time graphic that I have here on the left, and you can see that they're all kind of happening at definitely different rates. And if you are using this first order, which one do you pick? If you pick the first process, the fastest one here to model it, you have to do that anyway because you need some data. But let's say we pick orange, and orange is the fastest one here. Is it really going to capture what's happening in the blue? And is blue influencing orange? And we're not able to capture that. So those are some really big takeaways from just doing this first order modeling. Also, it takes much longer than a minute. So we have to wait for that fastest process to happen, but that still takes time. So you can't really do it in that amount of time. And I will show you some data to support what I'm talking about. And the results from this method are generally very unsatisfactory because they aren't consistent. Also, I'll show you with data. And here it is. So we did a study, we did a competitor study in our lab with our instruments and comparing with competitor A and competitor B. And show you here, we have different samples, and we tend to pick, these samples tend to be a little bit more difficult to test out. And we were looking at here the water activity. So there's the water activity for each one of these. And for each of the instruments. And then we looked at the time that they took. And I want to make sure I let everyone know that these were brand new instruments. They were verified and reading very well. They were clean, they were warmed up, everything was ideal. Even the sample was the same. So we just moved a sample between them so that we could, only thing that would be different would be between the modes. And you'll see here that for meter, we're always in equilibration mode, but for competitor A and competitor B, they have both a fast and an equilibrated mode. And so we're switching between the two modes. So this is just a comparison of those modes. Everything else was just the same. So I'd like to highlight some of the data here. So I'm going to focus on 3. And I want to point out here with the potato chips tend to be very difficult to read. They're very low water activity, plus they have a real high fat content, and that tends to limit that moisture mobility through the sample and then out into this and headspace. So they take a long time to read. So I want to point out here with competitor A, we've got a difference between the fast and the equilibrated at about 0.03 for that one, and which is fairly significant. And for competitor B, I want you to notice that it was better. It was like 0.01. But the other thing that's really kind of interesting here, if you look at this, and this is why this type of modeling is a little unpredictable, for competitor A, that water activity value went down. So the first reading was high, and then the equilibrated reading was low. And the exact opposite happened with competitor B. It came in lower and then went up when we let it go to equilibration. Next, I want to look at this ground black pepper, which is a really fine powder, and it's very staticky, and that gives it some difficulty. This one, the changes between them are a lot more extreme, and you'll see where they start and where they end are very similar between the competitors and even with Meter. But you'll notice this is a huge difference for this one. They start out for competitor A has a difference of 0.05, and for competitor B, the water activity, it goes down by 0.06, and that's really, really large. How do you account for that? We haven't, if you look at the rest of the graph, you can't see that much change, but for this particular sample, it's a huge change. And then lastly, I want to talk about the dog kibble. And you can see actually they did pretty well between their fast and their equilibrated states. There isn't much difference between them. A little bit. There's a little bit. But I want you to look at the times. So when you look at competitor A and the time it took to do the fast, it took over 14 minutes. That's not particularly fast. And you can see with the rest of them for competitor A, it's quite slow, really. It's an improvement over the equilibrated one most of the time, but not always. And when you look at competitor B, you can say, oh, look, that only took 3.8 minutes. That was pretty fast. There is a difference by 0.06 with that one. And if you go over and look at our primary method with the chilled mirror, you will see that it is equilibrated value, and it actually took less time in 3.2 minutes. All right. So what do? We can't really use that first order modeling. It doesn't work very well, and it's not very predictable. So we're moving to a second order model. Now, this model is developed by physicists, and it uses strategies from predicting environmental modeling. It's going to require sample-specific coefficients. When we see the differences between like the black pepper and the kibble and the potato chips that we were just talking about, you can see that all of those come to equilibration much differently from one another, and depending on the instrument too. And so we're going to need to be able to identify the specific sample so we can get the correct coefficients. And also we need some data. So we need 3 readings of that specific sample to start getting this model to get going. So this last graph I have here shows how well we did essentially. So let me explain it a little bit. We're going to compare the fast water activity, the 1-minute water activity readings, the air, the maximum air. How far off were we is essentially what we're looking at. And we're going to compare them to our final equilibrated dew point reading and the air involved just in that method alone. Now, when we started this, we were hoping for an accuracy of 0.02, which is what I have here at the top of my graph. Let me explain my axes a little bit. So the x-axis, we have the expected water activity, what we thought that the water activity should be. So for standards and for actual food samples. So we tested all of it, and we did it from a range from 0 to 1 for water activity. So we did the whole entire range. On the y-axis, we have the water activity maximum air. So how far off from that expected water activity are we? How much were the readings? And this is maximum air. So this is not average air or minimum air or any of that kind of thing. This is a maximum air for those readings. How bad could it be is really what we're looking at here. So now in the blue, we have our 1-minute water activity readings, and we're comparing them to the dew in the gold, like I said. And you can see that except for one sample, which is around 0.984, we were able to have the air, max air, of 0.01 or less. This is really amazing. So we do have the air spec at O2, but we can see here that we're always, almost always much under that at 0.01. All right, I'll turn the time over to Zachary now to learn about reducing uncertainty.
All right, thank you, Mary. Okay, so one thing that I wanted to highlight here is that 1-minute readings are possible because our normal readings are already fast. If you look back at the data that Mary supplied in that table, normal readings using our dew point sensor are often just as fast as some of the fast mode readings for some of our competitors. So these readings usually take a couple of minutes, somewhere around 5 minutes. The other thing is that these 1-minute readings are possible because we have extremely clean instruments. So again, like Mary mentioned, we have done a lot of research on eliminating materials that will adsorb water and looking at the different surfaces within the chambers for our water activity meters. The other thing is that we already have extremely accurate instruments. So our instruments can already measure down to 0.005 or 0.003. And because of this accuracy, second order modeling can then be applied to make these measurements even faster. So overall, if you have a sensor that is slow, if you have that slow sensor, then you can never get a 1-minute reading. But because we have fast normal readings and clean instruments and accurate instruments, we are able to use that second order modeling. So that this means that for us, variability is a bigger issue than accuracy. So this graphic to the right just represents different runs of the same food product. And within those different runs, you're going to have varying degrees of variability for water content or for water activity. And this variability is somewhat unpredictable. It doesn't necessarily follow a bell-shaped curve, and it can be different for each run. So this means that we need to take more measurements, and by taking those measurements, we are able to reduce uncertainty better than increasing the sensor accuracy. And I have an example here on the next slide. So this is showing how uncertainty is reduced for these water activity measurements. So let's say that we have a food sample. You can pick whatever food sample fits your application. And in that food sample, there is going to be an inherent variability that makes uncertainty high, especially if you are only taking one water activity measurement. So let's just say that taking this one reading for water activity generally takes you 10 minutes. But if you're using 1-minute water activity, in that same 10 minutes, you could be getting 10 readings for that sample. So let's calculate the uncertainty for both of these different methods. So as a reminder, uncertainty is equal to the standard deviation divided by the square root of the number of readings. So for the first example, if that uncertainty is 1, just to keep things simple, if that uncertainty is 1, then that means that using water activity, using fast water activity, that you could reduce that uncertainty to less than a third of the original uncertainty because you're able to take so many readings within the same amount of time. So fast water activity allows you to include more readings in your average to drastically improve your uncertainty, as we just saw in that last example. So if you can just get 4 readings, that will cut your uncertainty in half. Something that we see time and time again is that the main source of uncertainty in factories is not taking enough readings. A lot of factories will take a couple readings, and this is because they're generally limited by time. It can take a long time to get water activity measurements. So that is leading to lots of uncertainty in those measurements. Also, the inherent variability in the product means that 1 or 2 readings simply is just not enough to characterize the water activity for that batch or for that run. So overall, the more readings you can get, the better you're going to be. So now let's move into some of the business impacts of fast water activity. These are some feedbacks that we have already received from some early adopters of this technology. So some things that we've heard back is that companies are able to set up more robust testing protocols, and they're quickly able to get outside operating limit indicators. And that's because these fast readings can come with a quick indicator that we use, green or red, to quickly allow the operator to know if that batch or if that run is within spec. Other companies have told us that they're able to make far faster decisions at the line. This is helping to eliminate reworking and scrapping batches, and this is helping some of our clients to clear shipments at 5 times the speed that they were doing before. Also, having this technology is helping to eliminate any bottlenecks that may occur and to optimize logistics. So we're going to go through a couple examples We were working with a cured meat manufacturer, and now they are clearing their ovens and their cure rooms in a fraction of the time that it was taking them before. So they've actually been able to see a 40% increase in production throughput with no additional investment. And this was really helpful to this client because they had planned on spending about $1 million to scale up their operations, but by optimizing their current equipment and by applying this technology, they were able to drastically improve their throughput. Another company that was an early adopter was making oven-baked oat bars, and they were required to take 6 readings per run, and this was taking them over an hour. So they were able to reduce that time from over an hour to less than 10 minutes. And in addition, they could know with certainty when they needed to move to their packaging steps, and they were having no unnecessary rework or lost batches by using 1-minute water activity. Another early adopter was an innovative sunflower seed butter producer that we've been working with. They were actually having tests that were taking around 30 minutes. So imagine being able to take that 30-minute test and reduce it down to 1 minute. So now they are no longer having delays on releasing their batches. So from here, I just want to leave you with a few key takeaways from this presentation today. So let's start off with some insights, and those insights are that speed changes everything. So if you're with a company where speed is really important and getting those water activity measurements as quickly as possible, this technology is for you. The other thing is that moisture issues represent a cost. This is due to scrap batches and rework, and also moisture issues represent an opportunity because over drying can significantly impact your yields. So the benefits of fast water activity is that you can have greater control over your water activity, and time again, we have seen that this can lead to 6 to 7 figures in increased revenue. The benefits also include being able to make quicker adjustments to machine settings. So this gives you much greater control over your production processes. And this overall will allow you to deliver a consistent product with a spec that is tied to quality and safety. So some of the people who will benefit from this type of technology include manufacturers of shelf-stable intermediate foods. So this may include things like ready-to-eat meat products or pet foods, energy bars and snacks, fatty snack foods. For example, Mary gave the example of the potato chip. Companies that work with dried fruits and nuts, and also confectionery companies, particularly gummies, which can take a very long time to get water activity. Some other companies may include producers of jams and jellies, cannabis, seasonings, ingredients, basically anywhere where moisture is causing serious quality issues and you really need to dial in the water activity. So Mary and I want to thank you for your time today, and we're going to go ahead and move into the question and answer section. So I'll turn this back over to Brad.
Awesome. Thanks, guys. Let's take a few questions, and we'll get to as many as we can beforehand. We've got a bunch of questions that have come in already. I do want to let you know that we probably will not be able to get to all of your questions. And if we do not get to the questions that you have submitted, we do have them recorded, and Zachary or Mary or somebody else from our Meter Food team will be able to get back to you directly to address your question. So feel free to submit any questions and we will get back to you. Let's see. There's Zach, Mary, there's a bunch of questions on the modeling process itself and trying to know how well your model is working. So are you working with actual data? Is this theoretical data that you're putting out there? Also, with those the graphs of distribution of variability that you had, I think, Zachary, how is it that you can know that your model is working well?
Sure, I'll start with that question and then I'll let Zachary kind of finish it up. So far as the testing in the lab, we've been testing this from the get-go. We're always very concerned and wanting to make sure that our process is modeled. So it's been an evolution as we've gone through this. This data we've collected in the lab testing lots and lots and lots of samples. And like I mentioned before, we like to test the difficult ones. The easy ones, what's the fun in that? So we test difficult samples, like Zachary was talking about, things are slow to move that water vapor through, that are going to have long read times and things like that. So when I show you the graph of the max error, that's a condensed version of all of the data, the actual real data that we've been gathering in the lab. And to know if it's fitting very well, so when you take a reading, it takes 3 readings to be able to basically characterize your sample. And then you can actually you can take the 1 minute water activity reading as is. You can also let it go to completion and you can compare the two. So you can get a direct comparison of how well your model, how well it is modeling or characterizing your sample from there, and get you'll know from there. Was there anything more about that part of it for me? Sorry, I'm trying to remember. It was a long question,
Yeah.
Brad.
Yeah, there was a lot there, Brad, but I do think one of the key takeaways is that this does take some setup to get going, and that you do need to run your sample the full length of time at least 3 times, and that helps us to get the best prediction possible. Also, we can make a better prediction with the more data that we collect. So this is only going to get more accurate over time. And like Mary showed, even though the spec is for within 0.02, most samples are within 0.01 according to the data that we've collected.
A bunch of people here have been asking about in practicality in their use, how do they get this? How do they need to upgrade? Is this a software upgrade, firmware? What do they need to do to be able to get this 1 minute water activity reading?
Sure. So this 1 minute reading comes with our Aqualab 3. This is a new instrument. So there's no upgrade available for older instruments. But like I mentioned before, generally our clients are keeping their current equipment and continuing to use that in their lab, and then applying this new technology in line or in process where it is needed. So this goes with our Aqualab 3.
And I guess also the process of doing this, are they going to have different instruments, one running the actual readings, one doing the predicted reading? Can they get both the predicted and the actual reading on the same run, the same instrument?
Go ahead, Zachary.
Yes, you can get both those readings on the same instrument. So again, like Mary said earlier, you can allow your sample to come to the full length reading and then compare that reading to the 1 minute reading. So you can use the same instrument, the Aqualab 3, to get both.
Okay. Let's see. I think we'll have time we're running short on time. We'll try to get one more. Again, we will be getting back to your questions. There's a bunch of questions that came in. We've got a couple dozen here that we will not be able to get to. And somebody from our Meter Food team will be able to get back to you and address your questions directly. I guess, Mary, let's talk about how have you built up to this? What has been the process that you've used in the lab with research and development on this model? What has been, I guess, the pros and cons to developing this process, this 1 minute reading?
Right. Well, right off the bat, when they first were talking about how do we, wanting to do a 1 minute water activity reading, my first gut was like, well, samples behave differently. They equilibrate differently. Some take longer. Some it's very complicated because every sample is different. How are we going to overcome? How are you going to come up with a generic model that's going to account for all that stuff? And that's just what I was talking about when I look at it from a first order modeling perspective. It's impossible. You can't really account for all those things because they also influence each other, and they influence each other differently with different samples and different under different conditions. And so I can't talk about the actual mathematical model that's actually used. I can just tell you in general where it comes from. But that was a real breakthrough, you know, because that was the major question of, okay, we want to do in 1 minute, but we've got this big barrier. How is there a modeling process that we can use that would account for all of these? And they found one, and they, like I described, it comes from the modeling environmental conditions, and they were able to apply that. So then it is, okay, well, we've come up with that. That's a great idea. Does it work? And so we've spent so many hours testing in the lab, an iteration and iteration and iteration to get this tool down to what we've presented today. So it's months worth of work and months worth of testing that's been compiled into that. And so far as how it works, it's pretty impressive. When you set up the testing, you set it up by particular sample, and then you would give it some kind of quantifier for like a batch or a lot or something like that for your testing. So you kind of set up the testing in an overall system, and then because you've already specified what sample it is, it already knows. It knows all the history that's been included with that sample. So after you get your first 3 readings, and if it's a brand new sample, then you know it's just immediate, the first time you set it up and put that in, and then that's it. You don't have to do it again. And so you take your first readings, so they're going to be the same like the time that they would be. They're going to come to equilibration because we have to characterize that, and then you're done. And then you're in your 1 minute. And I also wanted to say, I mean, we do have the specification for the system that is 0.02, and that told you that's what our goal was when we were starting. But the fact that we actually met that in half, you know, at 0.01 for almost every sample that we tested, save 1, is really encouraging. So we have really good accuracy with this. The fact that we can do it just helps. I mean, the data proves the model. That's how it works.
So it looks like we're about out of time, but I do recognize a lot of the names that are attached to some of these questions coming in. And so Mary and I will make sure to reach directly out to each of you and to answer your questions. Thank you again for tuning in today. We really appreciate you listening to us. So thanks again, and we hope that you enjoy the rest of your day.
Yeah, thanks a lot. We hope that you enjoyed this discussion. I know we did have a lot of good in-depth questions that came through that are a bit too much for a quick discussion right now. But we will be able to get back to you. In the next couple of days, you'll be receiving an email with the links to the recording and the slides. And also, if you're interested in more webinars on shelf life, be sure to let us know in our post-webinar poll. Again, stay tuned for future Meter Food webinars. Stay safe and have a great day.